Hey, hi, hello guys. I hope you guys have been going through the lecture videos, and yeah, I'm just gonna keep accepting people. But in the meanwhile, if you guys have any questions from the previous sessions or any doubts, please do let me know now. If there are any issues in solving the assignment questions from the previous weeks, you can let me know now. Am I audible? Is my audio clear? Could someone please confirm?
Yes, ma'am.
Yeah, thank you, Sunil. Sunil, I think you also had some issue with completing the assignment. Were you able to get any response from the NPTEL team?
No, ma'am, I haven't got any update yet.
Okay. Uh, I can't really do much about it because they don't give us that access either. But I can maybe try writing to them again separately to see if they can just enable it for you. But I'm not sure if it's possible to bring back an assignment that was over in the previous
No, ma'am.
section.
I just needed to know that will I will be eligible to qualify 40% internal exams
You
that
can continue writing
writing.
the other assignments. I think that should be okay. If you
Yeah.
just miss one of the assignments of the rest of them, I think it should still be okay.
Okay, that won't be a problem, ma'am. That was the only major concern.
Okay, okay. Are you guys able to access all the YouTube video links and the presentation slide links and all of that? Is there something that you guys want me to add more to these sessions? Uh, if that would be helpful, I can try adding more information to these slides. If there's anything like that, please do let me know in the comments so that I can get back to it. Yeah, it's still like I'm gonna wait for another 3 minutes so that more people join in. I think more people will just anyway join in the middle, so I'll already share the screen and start with the session. If there are any questions, please do stop me in the middle. Okay, let me. Yeah, so my screen's visible, right, in PowerPoint mode. Could someone please confirm? Yes, okay, thank you. Yeah, so if there are questions, please do unmute and speak because I won't be able to see the chat box. So please do let me know if you want me to stop somewhere and explain it better. So this is the fourth session. So this is a total of a 12-week program. So we are in the fourth session now, and we have seen multiple basics of what environmental biotechnology is about, why studying microbes is important, and we have pretty much looked more about microbes in the last two sessions about their ecology. And the coming few sessions would be dealing with the methods, the techniques, the engineering techniques, what are all the different methods to assess the microbes themselves, to classify them, to identify them, to engineer them, and so on. So, this session will also focus predominantly on the environmental engineering part of it. So we had already briefly seen some of this thing in the previous classes about how do we assess any environmental sample. So this could be a water sample, a contaminated water sample, a contaminated soil sample, or any of the environmental sample could be assessed for the presence of microbes. So here this is from a particular paper where they have looked at the different aspects of a contaminated soil sample. So why are they looking at a contaminated soil sample is because this will give us an idea about different things, the different kinds of microbe that are there, the microbes that are that could be useful in degrading the contaminants, how the microbes are in general growing, how it has affected the natural diversity, and so on, can be assessed using some of these techniques. The major techniques are genomic and metagenomic techniques. So all of these are metagenome, metatranscriptome, metaproteome, and metabolomics, and so on, because we are going to collectively assess all the bacterial communities together. So it's not for a single species or a single strain of organism. It's for collective communities of microbes that are present in the environmental sample. So this is that's why it's metagenomics. So here we can assess the DNA of these individual organisms, their genomic uh, 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 elements, the elements, genomic elements in their organisms. Now the genomic techniques are so good, and the bioinformatic have evolved so much that now we can analyze the whole genome of microbes at ease. So we can look at how they are evolving, how these contaminations have changed them, if there are any mutations in these organisms that is reducing their diversity or increasing their numbers, and so on. So DNA is one of the um, the places where the signatures of the environmental destruction could reflect. We can also look at the transcriptomics. Transcriptomics is the mRNA profile from all of these microorganisms. So all the organisms convert the genomic information into mRNA, which gets translated to proteins, and proteins are involved in multiple pathways that produce different primary and secondary metabolites. So transcriptome is like a snapshot of what exactly is happening at that particular time point when you collected the sample. If some of the genes are upregulated, if some genes are downregulated, if some kind of microbial interactions are happening because of the contamination, and all of this can be seen in the transcriptome itself. The next thing is the proteomic signatures. Just a second. Yeah, and the most important thing is the proteomics, because this is a lot of things can get translated, can get transcribed to mRNA, but there could also get silenced in the process before translation. So proteomics it represents the exact output of what exactly these microbes are doing, how they are behaving in the environment, and hence looking at the protein profiles, the enzyme expressions, or the different proteins produced by the microbes will give us an exact idea of how they are responding to a given environment. So it gives us an idea about the bioprocesses, and it can also help us in characterizing the functional molecules. Metabolomics is the final end product. So this can give us an idea about the metabolic pathway in the microbe itself. This can give us information about the different kinds of substrates that they are consuming, what are all the end products that are being converted by these microbes, how they utilize energy, how we can use them for generating alternative source of energy, what is the end product that is formed from the metabolic processes, whether it is harmful to the environment or harmless, and all of this information can be obtained from metabolomics. So all of these are different bioinformatic tools that will help us in understanding the nature of the microbial community in the contaminated region or versus their natural habitats. But what are all the challenges involved in understanding these different processes? Apply these methods, most of these methods that I described so far, the transcriptomics, proteomics, all of these are developed for single organisms or a single species. Trying to do this at a meta level for a whole microbial community is the main challenge. Because these techniques themselves generate a lot of data inherently, now this data is going to be multifold because we have a whole microbial community to be assessed. So yeah, maintaining and assessing this large amount of data requires lot of computational power, which is not very easy to be done everywhere. And not just collecting the data, we can collect lots and lots of bioinformatic data with the current tools that we have, but obtaining some meaningful information out of the data, processing the data, and then understanding the theory driven concepts. So if we have a lot of data, it is about putting them together to get an idea based on the theories or the principles that we know about these microorganisms, is the most important challenge with dealing with such large data. And also upscaling the microbial process for application. So whatever we are looking at would be at a very small scale. So we'll be taking a small microbial sample from a soil sample, and we'll be looking at what exactly is happening. But if we do figure out that there are some enzymatic activities that are helpful for us in further applications like degradation of certain contaminants, then the problem is about upscaling. How do we upscale the level of the processing that can be done by these microbes for decontaminating an whole area, like a whole contaminated soil region, or decontaminating oceans, would mean that we need to upscale these microbial processes. And these whatever processes that we develop should also be reliable and cost effective. Yeah. So the practical goals of microbiology of engineering systems. So we do get a lot of data. So what exactly are our goals? What are we looking at? Uh, we have gone over this multiple times earlier as well. Microbial treatment of wastewater, soil, solid waste. These are the most important things that we are looking at. So whatever our data is going to give, this is what should be the end product. It can also be used for developing methodologies for prevention of diseases. We do see a lot of disease outbreaks that are happening in the recent past, so we want either something to do with environmental treatment or for prevention of some of these disease outbreaks. And the most important thing is also for controlling and monitoring these systems itself. So do when we do get the data, how are we gonna control and monitor these systems once they are out there in the actual application process? So once we apply them to the soil, how are we going to control them from evolving further into something that is not desirable, and how are we going to monitor these microbial systems? So that is also one of the primary goals of microbiological engineering systems. So what are all the requirements for achieving these goals? So we need to understand how we classify them, and then we need to understand the different interactions between these different organisms. This is a classification of an environmental sample. So these, as you can see, these are the different kinds of microbes that are present, and it is important for us to classify these individual microbial communities into their respective phylogenetic positions. And the second most important thing is to understand their interactions. What kind of tropic interactions do they have with themselves or with the other organisms, about syntrophy and competition and so on, and also the signaling mechanisms that they use for this kind of interactions. How do they sense each other? Does it happen through metabolites? If there are metabolites, what are all the kind of metabolites? So do they signal through antimicrobials, or do they give transcription based responses? So these are some of the things that we need to understand to effectively use them for the goals that we had described in the previous slide. And the third important thing is also to put together all this information to decipher the biochemical and the physiological adaptations themselves. So if there are so many number of microbes present in a given soil sample, for example, how do they relate to each other? And then we are understanding their interactions for that. But then what happens during those interactions? What are all the biochemical molecules that are released? How do those biochemical molecules influence the production of other biochemical molecules? So these are some of the things that we should be looking at to chart out or make a map of the different biochemical and the physiological adaptations that these microorganisms have. So these are the three major requirements that we need to get from our data uh, to understand and achieve the goals of environmental engineering. So this diversity that I spoke about, this is something that's very basic that we need to understand. Any microorganism will require an energy source and a carbon source for its life cycle. And this, based on this energy source and the carbon source they use, we can classify these organisms, and this is a classification based on their metabolic diversity. So this classification is not based on their taxonomic classification like their genetic information and so on, but this is based on how they produce metabolites and what are all the sources that they use. For instance, we will also read little bit more in detail for many of these subtypes in another chapter. But here, to just get an overall idea, photo means light. So we have words like photosynthesis, photography, and so on. So photo means light. So here, this word, the nutritional type which classifies these microorganisms, is a combination of what are they using as an energy source and what they are using as a carbon source. So photoautotrope is an organism that uses light as an energy source, and auto is an organism that that produces its own food and does not depend on any other organism. So carbon dioxide directly is the carbon source for this organism, and thereby a photoautotrop uses light and carbon dioxide. So this is all plants that we see around us. Many of them are photoautotrophs, and some of the microbes, including cyanobacteria, green, purple bacteria, all of them come under photoautotrophs. Similarly, we also have photoheterotrophs, chemoautotrophs, and so on, based on the light source. So, if they use organic compounds as light carbon source, they are considered as heterotrophs. And then heterotroph is something basically that depends on some other organism for obtaining the carbon source. The classification also changes depending on whether it's an organic compound or an inorganic compound, and the energy source, if it is a chemical compound, then that they become chemotrophs. So there are more classifications among the organisms like that, but the whole idea here to understand is that whatever organisms that we are identifying in our environmental samples need to be classified into these different nutritional types, so that we can get more information about what kind of energy source and carbon source they require for growth and for the further applications. And they can also be classified depending on the conditions that they require for growing. They can be aerobes or anaerobes. Anaerobic is a condition where they grow in the absence of oxygen. So again, we can again classify microorganisms based on whether they are aerobes or anaerobes. Obligate aerobes are organisms that can survive and grow with the presence of oxygen. The complete opposite is an obligate anaerobe, which cannot live in an oxygen containing environment. Just the presence of oxygen itself will lead to cell death. So this is the basic between an anaerobe and an aerobe, whether they can live in the presence of oxygen or not. But there are also few other types of organisms which are not completely on either of these sides. For instance, facultative anaerobes, they can live between aerobic and anaerobic condition, and they can switch between aerobic and anaerobic condition depending on the availability of oxygen. So they can actually grow in an anaerobic condition and an aerobic condition as well. Aerotolerant anaerobes, on the other end, are oxygen tolerant. They can't really use oxygen for metabolism, but they don't really die in the presence of oxygen. Like obligate anaerobes. Obligate anaerobes just can't handle any oxygen at all. So if you put them in an atmosphere containing oxygen, they'll just die. But on the contrary, aerotolerant, the word tolerant itself means that they can tolerate some amount of oxygen. So they may not be able to grow and produce metabolic components, but they will not die. So this can give us some buffer time to transfer them to an anaerobic environment. And then there are micro aerophiles, which need oxygen to grow, but at higher oxygen concentrations they'll start dying. So they don't live in an oxygen rich environment, but they grow in an oxygen containing environment, as opposed to obligate aerobes. So yeah, these are the different ways we can classify the microorganisms that we obtain from our environmental samples. Now we're going to look more about the different kinds of microbial organisms that we will be getting from our environmental samples. How do we engineer them and so on. But before I get into that part, if anyone has any further questions, or if there are any comments, uh, right. Are there any questions? Okay, let me just go back. Yeah, so fungi is another microbe that usually is seen growing in most of the contaminated sites. They are often seen near biomedical waste, agro waste, mining sites, and municipal waste. You can just see that fungus notoriously grows in all of these environments. But how do we understand them and classify them as which are toxic, which are non-toxic, which can be used for further applications and so on? We can collect the fungi, obtain a culture, and then do various screening tests to understand what kind of substrates they grow on. So that the kind of fungi that is growing in a particular area can give us more information about what kind of waste is observed more in these regions. So if a particular fungi is growing near a region which generates a lot of biomedical waste, then we will be able to understand what kind of harm this waste contains, what kind of pollutants this waste contains. So that is a way fungi can be used for understanding the kind of substrates that are available in a contaminated site. It can also be used for understanding their heavy metal binding ability, because a lot of fungi have heavy metal binding ability in their cell wall, and hence they can be used in zones which have a lot of heavy metal contamination. So this kind of different degradation and absorption studies can be conducted on fungi that are isolated from an contaminated environment. There are different groups of fungi, and various representative members of the fungi family are given here. There are zygote producing fungi, sac fungi, club fungi. These are just the ways, different forms in which they grow, and each of them have different advantage. Some of them reproduce asexually, and some of them reproduce through sexual reproduction. And this gives us an idea about what kind of an adverse environment can they grow on, whether they will remain as spores and then grow on later on when the conditions become favorable for them. We can also understand whether they are unicellular or multicellular, what kind of mechanisms they use for asexual reproduction, the different kind of vegetative reproduction, budding, and so on. And some of these fungi are also human pathogens. So understanding them and classifying them like this will give us an idea about what are all the different contaminants are giving rise to what kind of different fungi. The second most important organism that we need to know about, especially while studying contaminated samples such as soil sample and contaminated wastewater, is algal samples, because algal growth is what we observe in many of these contaminated regions. Algae use sun's energy directly, so they are photoautotrophs. They produce their own food. They have chlorophyll just like plants, and they produce their own food, so they take in carbon dioxide. So because they take in carbon dioxide, they produce a lot of secondary metabolites, including biofuel. They are a great a way of utilizing the existing waste. The waste can be in the form of the carbon dioxide waste that we generate, and they can use that waste products to convert it into some of the best usable products, such as biodiesel and so on. Therefore, algae is one of the main organisms that we might have to understand in detail, especially from a contaminated region. But how to grow microbial biomass? The microbial growth yield coefficient. So this is something that's empirical. We do not have any derivation or formula for this. So any substrate in the presence of oxygen will be converted into products and biomass by the microorganisms. And biomass is it could be a result of aerobic process or anaerobic process. But this formula CH1.8 O 0.5 and N 0.2 is, like I said, is empirical and does not have any derivation. And this formula of the biomass can vary between different microbes. So we have to individually calculate depending on the amount of substrate and depending on the amount of oxygen available to see what is the formula for the biomass from our particular reaction. And in any microbial reaction, the average carbon content will be around 50% of this total biomass. If a particular microorganism is producing, let's say, X percentage of biomass with the help of substrate and oxygen that we are providing, then the 50% of the total biomass will be of that of carbon. So why do we have to study about the microbial biomass production and the carbon content and all of that? Because this is one of the major things that will give us an idea about what kind of biomass that the microbial processes are producing, whether whatever that we have in the end can be further broken down into harmless products for the environment, or whether this biomass can be further utilized for another alternative source of energy and so on. Hence, understanding the percentage of biomass and the constituent of different elements in the biomass is one of the most important steps in understanding environmental engineering and scaling them up for a larger area. So this is actually my last slide, and we are only like half an hour into the class. This week's lecture session mainly had many of these things, these ideas, especially about the different microorganisms that we need to understand about, fungi, algae, and different kind of microbes and so on. So these are more like theoretical. So if you guys have already gone over the lecture slides and have more questions about what exactly is important in each of these slides, we can definitely go back to some of these things and discuss. We can even discuss more about their classification or how some of these techniques are done. So in case you have any questions from these previous slides, please do ask me now. So yeah, so this is actually just the last slide. So this is about how the different substrates are utilized. So this is a snapshot from the lecture slide itself. So this is a substrate, and x1, and x1 is the component that is produced. So this varies depending on the different substrates that we give for the microorganism and the respective biomass that is produced. A generic equation is written in case of oxidation of carbohydrates to generate energy, which can be used for growth. This is how the generic equation looks like. Whereas in the case of oxidation of carbohydrates to reducing equivalents, so here this is an oxidation. So here, in the presence of oxygen, it is getting converted into energy and biomass. Whereas in this particular case, there is a reduction happening, which is further used for biomass synthesis. So redox reactions, which we will again look in detail in some of the future coming sessions, is the main reaction that happens in any of the substrate utilization reactions of microbial action. It is one product that is oxidized and another product that is reduced, which results in the formation of a final biomass. So in the case of an oxidation reaction, we'll see that oxygen is being taken in by the substrate to produce the biomass. Whereas in the case of an reduction reaction, we see that hydrogen ions are produced along with the energy. So this is the oxidation of carbohydrates to generate reducing equivalents. And the third is the common form of biomass production that we see in many of the cases. This happens in fermentation reactions. This happens in several microbial reactions where assimilation of carbons from the carbohydrates take place for production of biomass. And this, as you can see here, is also the so X here indicates the balancing values. So depending on what is the substrate and how much of oxygen is required, the unit of X will change. So how many units or how much of oxygen is required to convert a particular substrate, and how much of carbon dioxide is produced. So this is basically just a chemical balancing equation where the X is a numerical unit which will keep changing depending on the number of units of carbon and oxygen, or the substrate, or the final carbon dioxide production takes place. So this is the empirical equation that we saw in the previous slide for any given biomass production. So this is a classical example of assimilation of carbons for production of biomass. So it can so these question these some of these slides are mainly designed to help you in solving the assignment questions, so you can get an idea about what kind how the equation will look like depending on what is the condition in which the substrate is subjected to. So actually, yeah, today I don't have more from these particular sessions. So in case if you guys have any questions from the slides that are there, or even in the assignment questions in your current week, you can post it here and you can ask me those questions. Do you guys have anything to clarify? Yeah, Sunil.
Ma'am, actually, I have out of the slides question.
Okay.
I just interested to know what are the laws and regulations for import-export of microbial strains? Are there any specific parameters, or
Yes, there are.
I just
Yeah, there are certainly. Because not we will see in some of the slides that not all microbes can be grown under all the conditions, and since their evolution, their microevolution depends on the other microbes around, you can't take a microbe from one microhabitat and later on release it in a different microhabitat. They might be producing. Um, uh, Substrate, I mean, they might be degrading substrate and producing products that could in turn become harmful to the other microbial species there. They can become an invasive species by using all the resources that are suddenly available to them. In one of the previous slides, we also saw about niche, about how a particular microbe plays a certain role in the environment. So there are definitely rules and regulations for importing and exporting of any living organism, and that applies to microbial strains as well, because it won't be a problem using them under controlled laboratory conditions as long as these are not released into the environment. I think doing something in the lab should be fine. So there will be laboratory biosafety committees which will assess whether these strains are fit to be used in a particular region, and how the waste management would be done, because managing the waste is the most important thing about using some of these strains that are not native to a particular region.
okay, ma'am. Are there any particular informations or the committee which are monitoring these? I just
Yes,
want
so
to know the names
this
just for the details.
usually is monitored by the Institutional Biosafety Committee, but there are also committees for the national level. It depends on what kind of strain it is, what is the biosafety level. So some strains like E. coli are have the least biosafety concerns, so they can be you can work with them in a biosafe BSL facility 1 or 2 itself. But there are certain microbes, including mycobacterium and other harmful pathogens that cannot be used under normal biosafety levels. So it depends on.
Algae is. What about algae?
Yeah, algae are not pathogenic to humans in any of the cases. Just that any samples that have lot of algal biomass, when they are released to wastewater, if they just mix with the normal sewage, they will result in eutrophication. A lot of algae will start growing. So you might just need to treat them properly. So anything, all of this can be taken care of by the institute or the place that you are associated with.
Okay, ma'am. Thank you.
Thank you. May I know your background? Like, are you from the industry, or are you a PhD student, or what? What
No, ma'am,
exactly?
actually, I'm a commerce student, but I'm just more into the science and aspect, everything.
Okay, okay. Because you're specifically interested in doing some of these
Yeah,
microbe or algae based. Okay,
microbial activities. It
okay,
was just going through a thorough knowledge. Okay, what are the things currently going on?
okay.
Yeah.
Yeah, okay, cool. All the best. Anyone else has any questions? So it doesn't have to be questions from these presented slides or from the sessions, like the lecture sessions of this week itself. It could be in general about environmental biotechnology, in general about biotechnology in general. It could be anything outside, and you can also ask more about genomics or transcriptomics itself if you're still not clear about how exactly to use those tools for understanding microbes or understanding any system of your interest. So we can discuss a little bit more about those techniques themselves. So did everyone understand about uh, the different things? Metabolomics, what exactly do we assess there? Genomics, transcriptomics, and proteomics, what kind of softwares or tools we will require. So if you have any of those questions, you can definitely ask those things here too. Most of the sessions are actually going to be small, because in the next few coming slides it's all techniques and methods based, so you probably just have to go over the slides and ask questions if any. um, I'm also again going to be traveling back to the same place that I was a couple of weeks ago in Rajasthan, so I might have network issues. So you guys can always write to me in my mail ID and ask questions in case if I'm not able to clarify some of these things. Um, I can also make, if you guys are interested, I can add a little bit more about uh, my own work if you guys would be interested in understanding this. I guess many of you are from here from the first few sessions, so if you know, I'm working on venoms, and today as well I'm in my lab. So uh, we use some of these techniques in our own lab as well, genomics, transcriptomics, and proteomics, to understand how venoms vary, what are all the different components of venoms, and so on. So if you would like me to extend these sessions to talk a little bit more about the application of these techniques into my own research, I can definitely clarify. And if you guys, if any of you are doing any research and want more information about how to apply these things in your research, you can certainly let me know. We have only like 10 people and we still have time, so maybe you guys can introduce each other and tell me a little bit about yourself so that I'll know uh, what exactly you guys are looking for in these sessions, so that I can uh, streamline the sessions to better to make your time worthwhile. So maybe I can just call out names and you guys can just quickly tell me what you guys do, or what you guys are why you taking this course mainly. um, I'll maybe start from the end. Yogendra, could you please unmute and introduce yourself?
Good evening, ma'am.
Yeah, hi, good evening.
Hi, good evening. Myself, Yogendra Chaudhary. I am from Alwar, Rajasthan. Uh, I doing my MSc in final year at Central University of Rajasthan in MSc Biotechnology.
Okay, so this course you're taking just for this is a part of your MSc or?
Uh, MSc, part of MSc.
Okay, okay, sure, thank you. uh,
Yes,
Sushmita.
thank you.
Yes, ma'am. Good evening, ma'am.
Hi, good
Myself,
evening.
yes, ma'am. I'm Sushmita, ma'am. I'm Dr. Sushmita from Usmania University, ma'am. I'm working as faculty in St. Pius College.
Okay.
As an Swayam course, I thought of doing this course when I gone through all the videos, and it is so informative, and it is moreover, it is relevant to the present situation, how
Okay.
we are going to be use environmental biotechnology. That name itself attracted me a lot. So I'm learning and I'm following. Today only I joined. Every day I thought of joining for these sessions, but I couldn't join.
Okay.
So today I thought I will join, but I joined late, but I can
Yeah,
understand.
the
And it was
sessions, yeah, the sessions are recorded and the links are available. Like they're there on YouTube, so you can always
okay. Yes,
go back
but
and look at the videos. Yeah.
yes, yes,
Thanks a
and
lot,
it is
Sushmita.
informative. Yes, ma'am. It is informative, and I'm enjoying all the sessions as well in the
Thank you so much.
Swayam
If
course.
you want anything more to be added, please do let me know.
Okay,
You can mail me also separately and tell me if there's
sure.
something specific that you want me to include in these sessions. Thanks a
Yes.
lot, Sushmita.
Yes. Thank you so much, ma'am. Thank you, ma'am.
Sunil, I think I'm gonna skip because he did already speak about himself. Next is Neha Sagar. Neha, could you please introduce yourself? Neha, you can you hear me? Okay, I'm gonna skip Neha. I don't know if she can hear me. Uh, Dr. Durai says knowledge enrichment only. Uh, yeah, sir. Please do let me know your comments if these sessions are um, if these sessions are good, and if you want me to include anything more into these sessions. I'll definitely be happy to include them. Yeah, I think there were a couple of other people who just left. So I think I'm going to just close this session early. Thanks a lot, everyone, again for joining. I only remember like a couple of people who join every week. Um, so, yeah, thanks a lot for joining.
Thank you.
Thank you.